Okay, so I was asked to talk today about analysis methods for large-scale multi-region recordings using Neuropixels probes. In the time I have, I can't survey all the work in this area, but I'm going to focus on the work that we recently published in this paper, which you can read more about on bioRxiv if you're interested. The conceptual foundation of our approach should be somewhat familiar after the last few talks, and it's the idea that cognition depends on signals that are internal to the brain. Um, what I mean by this is that cognitive processes like decision making, working memory, attention are poorly predicted from external events like a stimulus or a motor action. And this in itself is not a novel idea, but we think it's in multi-region recordings where this really takes off. Yeah, so they're not correlated with external events, but instead they reflect internal signals and states that we can observe by recording for many neurons. So here's a schematic illustration of what we can do with multi-region recordings. So let's say we're recording spikes simultaneously from 3 regions, which we believe represent some latent cognitive signals of interest in their firing rates. This cognitive signal is not evident just by looking at these spike rasters. Instead, by projecting neural activity in each region onto what we believe is some relevant subspace, that is, by multiplying each neuron's firing rate times some set of weights W and summing the outputs, we can infer the evolution of these latent internal signals from each region. And let's just say that this is what they look like. It doesn't matter what these represents for the time being. So when we do that, not just for this trial, but across many trials, we can see that the evolution of these signals closely covaries between regions A and B, but not region C. And if you look more closely, you can actually see that there's this continuous wiggling signal, which appears slightly earlier in region A, the red region, and then an abrupt state change when the original signal goes flat, and this state change actually appears slightly earlier in region B, in the blue region. So this tells us a lot. It tells us that the dynamics of this signal that we've decoded is not just noise at the level of single regions, but a real shared internal signal. And it tells us that regions A and B reflect some shared computation, while region C does not. And third, it tells us that region B is clearly not the source of the continuous signal, since it shows up there later. And by the same logic, that region A is not the source of this discrete state change. So multi-region recordings allow identification of internal signals and state changes with higher precision, and they provide constraints on the multi-region circuits that give rise to them. So we're going to use this basic conceptual approach in the context of decision making. And this is a great area to study coordination of internal signals, because we know that neural correlates of a decision are widespread throughout the brain, as has been alluded to in past talks. One illustration of this is that you can decode a subject's upcoming choice from neural population recordings in many different regions. But what we still don't really know, although the work you just heard from Gabe helps to answer this as well, the big question is, how is this distributed activity coordinated on single trials to generate a coherent choice? So this is the question we wanted to tackle, and we're going to do it with large-scale multi-region recordings. I have to tell you a little bit about the behavior. So we trained rats on an auditory decision-making task. The rat initiates a trial by poking into the middle of 3 ports. While keeping its nose in the center port, the rat listens to 2 randomly timed streams of auditory clicks played from loudspeakers to its left and right. Click, 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 click. And then it's rewarded for licking the side where more clicks were played. And so it has to keep kind of a running tally of the number of left and right clicks, accumulating evidence over time to make its choice. And we'll analyze neural activity during the period when the subject is forming its decision, but before the motor response is allowed. We developed new methods for large-scale chronic neuropixels recordings in rats. 8 neuropixels probes were chronically implanted at 4 locations mirrored bilaterally, letting us record from thousands of neurons and dozens of regions simultaneously across both hemispheres. Since this is an analysis talk, not a Neuropixels implantation talk, I'm going to skip any further details about the methods for doing this, but I'm happy to answer questions after the fact. So I'm going to actually just skip this slide. And so that's the data we collected. What I'm going to show you is that two internal signals dominate brain-wide decision-related activity in these data. The first is a continuous signal, which reflects the gradually evolving decision state. And the second is a discrete signal, which reflects the moment the animal makes up its mind and commits to a decision. And so I'll take those one by one, starting with the continuous signal. To measure each region's representation of the evolving decision, we did the following. We take a time-varying weighted sum of the firing rates of each unit, such that we could best predict whether the rat would choose left or choose right. And this gives a scalar quantity at each moment in time, referred to as the decision variable, or DV. One way to think about this is in neural state space, a high-dimensional space whose axes are the firing rates of each neuron. Here I'm just showing 2 dimensions of that space for illustration purposes. So a point in this space represents the instantaneous state of the population in one region, DMFC, on a given trial. And as time within a trial evolves, you can see that the population state tends to separate by choice as the subject forms its decision. The weighted sum on the left corresponds to projecting the population state down onto the axis that best separates the two choices. And we show this projection for one example trial in red across time points. The result of the projection is just the magnitude of this red line. That's what we mean by the decision variable, or DV. So here's the DV across several hundred trials color-coded by choice, again with the example trial in red. They tend to ramp in one direction on right choice trials, in the other direction on left choice trials. But as Gabe showed in the monkey data, there's enormous trial-to-trial variability in this decision signal. And our operating assumption is that this DV that we've decoded is a neural estimate of the evolving state of the decision in the animal's brain. What simultaneous recordings allow us to do is to observe how this decision variable evolves across many brain regions at once. So here are the DVs estimated from all recorded regions across 5 example trials in one recording session. And what I hope you can see right away is that the DVs are quite different across these 5 trials, but very similar across regions, similar within a column. And importantly, this is not just due to the shared influence of the stimulus or to motor preparatory signals. The identical sequence of left and right clicks was presented across this set of trials, and the subject made the same choice. So that means that this trial-to-trial variability has to reflect internally generated decision signals. Here's the average of all the single trial traces for each region. And just reiterating the point that Gabe made, many past studies have focused almost entirely on comparing these trial average signals. But with single trial data and with simultaneous recordings, you can see these shared signals on single trials that are completely missed in trial averages. To quantify the single trial coordination, we applied PCA, principal components analysis. Geometrically, you can think about this in a state space spanned now not by individual neurons, but by the DVs in each region. And PCA quantifies the amount of variance explained by the principal axes of the data in this space. Or to put it another way, it helps provide an estimate of the number of state variables needed to explain the data. What we found was that a single principal component was sufficient to explain almost half the variance of the data and was the only significant PC. The rest fell below the noise floor. So what this suggests is that these brain-wide decision variables reflect a single shared decision computation across the brain. Next, we asked, where does this shared decision signal first emerge? To answer this, we can compare latencies across regions. To do that, we calculate the correlation between DVs in each region as a function of different temporal offsets or lags. So here I'm showing the cross-correlation function for the DVs in M1, primary motor cortex, and all other regions. And you can see the peaks are shifted to the right of 0, indicating the decision variable in M1 tends to lead the rest of the brain. And of course, we can calculate the peak and the cross correlation function between all pairs of regions. And we can summarize the data by showing the average peak lag for each region relative to all the others. And what this shows is that M1, along with two other frontal regions, DMFC, dorsomedial frontal cortex, and ADS, which is anterior dorsal striatum, this is a set of interconnected frontostriatal regions, that they tend to lead the rest of the brain. So this tells us that the shared internal decision signal likely originates in a subcircuit containing these regions and is then globally broadcast across the rest of the brain. Okay, so I showed you that we found an internal signal reflecting the gradually evolving decision state. Next, I'm going to talk about an abrupt state change at the moment the animal commits to a decision. So decision-making often involves these two sequential phases: evidence accumulation and decision commitment. In the first phase, the subject is actively considering new inputs that bear on the decision. And then at some discrete moment, the subject decides they've made up their mind, they've seen enough, they're ready to make their choice, and they ignore subsequent input. And this transition is captured by a very successful model of decision-making called the drift diffusion model. Just to review what this is, we assume some noisy latent evidence accumulation process, Z, whose rate of change over time, Z dot, is affected by sensory inputs and noise. And pulses of evidence favoring one option push the accumulator one way, and pulses of evidence favoring the other option push the accumulator the other way. And when its absolute value reaches some bound at plus or minus B, it stays constant. It's absorbed at that bound and ignores future sensory inputs. So this bound crossing captures what we mean by committing to a decision. And of course, the sign of Z at the end of the trial determines the choice that the subject makes. Recently, my colleague, Thomas Luo, developed an extension of this model called the multi-mode drift diffusion model, which leverages large neural population recordings to infer the time of commitment on single trials. So this model assumes that the firing rates of a population of neurons are driven by this latent accumulation process Z, with each neuron having its own encoding weight, W. And importantly, the encoding weights are allowed to be different in the evidence accumulation versus decision commitment periods. Another way of thinking about that is that the direction in neural state space that the decision occupies is allowed to abruptly change at the time of commitment. And so with enough neurons, this model gives us the statistical power to infer the moment of decision commitment precisely on individual trials. We call this estimate the neurally inferred time of commitment, or NTC. And what our brain-wide recordings allow is observing how this is coordinated across brain regions. These tick marks reflect the NTC derived independently from different regions. So the first thing we can do, similar to what we did with the decision variables, is ask how NTC co-varies across regions on single trials. So here are 6 example trials, again, chosen so they have the identical stimulus and choice. And what you can see is that the timing of NTC varies widely across trials, but this variability is largely shared across regions, meaning it reflects some shared internal signal. And I should emphasize the animal is not allowed to move to report its choice until after this period is over, after the stimulus is over. So this inferred moment of commitment is an internal event happening in the animal's mind that's not directly linked to the stimulus or to the motor action. And then as we did with the decision variables, we can apply PCA to the NTC data across regions as a form of dimensionality analysis. And again, we found just one significant PC, indicating that NTC across regions reflects a shared internal state change. And as with the decision variables, we can also perform a lead lag analysis comparing the relative timing of NTC. And this again shows that M1 followed by ADS and DMFC, this frontostriatal subnetwork, leads the rest of the brain. The decision commitment signals detected first there. Okay, and the last thing I'll show you is we wanted to ask a broader question: what happens across the brain at the time of decision commitment. So similar to past studies, we can predict the subject's choice with increasing accuracy over the course of the trial from every region we recorded. But now we can detect on each trial when decision commitment happened. To avoid circularity, we'll just use the NTC estimate from one region, M1, and then look at activity across the rest of the brain aligned to that moment on each trial. And when we do this, we find that NTC marks a dramatic halt in the growth of choice prediction accuracy, even when the onset of the rat's response isn't until hundreds of milliseconds later. So there's this very abrupt brain-wide state change. In addition to this plateauing of decision-related activity, we also found that brain-wide decision signals dramatically decorrelated. So to quantify this, I'll return to the PCA analysis from earlier. Here I'm showing you the fraction of variance in the brain-wide DVs explained by the first PC as a function of time. I told you before that the first PC explained almost half the variance, but it turns out that this high degree of coordination is unique to this pre-commitment period. Just before NTC, that value drops dramatically and stays low for the rest of the trial. So taken together, what this shows is that decision commitment is marked by this coordinated brain-wide state change in which decision signals abruptly plateau and decorrelate. Okay, so to summarize what I showed you, I laid out an approach for analyzing multi-region data, which is to identify low-dimensional signals, which can be continuous or discrete in individual brain regions, and analyze spatiotemporal correlations in these signals across regions. Using this approach, I showed you that two internal signals that we discovered, which dominate decision-related activity across the brain. By analyzing the spatial and temporal correlations amongst regions, we showed that these reflect computations that emerge in a specific subcircuit and are then globally propagated across the brain. And more broadly, we think these results are just one example of how multi-region recordings can reveal internal cognitive signals that dominate neural activity during cognition and which help us place novel constraints on the multi-region circuits that generate them. So I want to acknowledge the collaborators who contributed to the work, and I'm happy to take any questions.